This is TWIS. This week's Science episode number 1037, recorded Wednesday, November 12th, 2025. Science and Wine Roulette. Hey everyone, I'm Dr. Kiki, and today we will fill your head with supernovae, horny bees, and lady brains. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience. Disclaimer, disclaimer, disclaimer. This week is like none other. Come one, come all, enjoy the show. Science is always a stunner. Here on This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science, everyone. I can't say anything else. It's everyone out there because I'm all alone. I'm Dr. Geek here talking about science on This Week in Science. Thank you, everyone, for joining us for this episode. It is so fantastic to have you here. I hope that you are all enjoying the week. I hope that we are optimistic about the future and that we are doing the best that we can, because that's all that we can do, right? And have a little curiosity about the world around us. So that's what we're here for today. I would ask my co-hosts what they brought to the show today, but they're not here. It's just me. That's it. Just Dr. Kiki talking with you this week about the things I thought were pretty cool. And so the title of the show, Science and Wine Roulette. That's because I'm here with you, drinking wine as I enjoy doing. And it's kind of a roulette and an AMA and talking about science tonight with everyone who's in the chat room. So if you're in the chat room and watching this live, know that I'm here to answer your questions. We're going to have a good old-fashioned science chat, and I am really looking forward to hanging out with you for, tight 90, maybe less, maybe more. We will see how it goes. In the meantime, for those of you who are interested in what kind of stories I actually brought this evening, whether or not we actually get to them, I have stories about malaria drugs, Epstein-Barr virus, and autoimmune disease, the way that ice raids are interrupting education, a couple of supernovae stories. I mentioned a horny bee. Yes, I've got some devilish bees. And let's see, some brainy stuff, lady brains, fixing brains, organoid brains, and kid brains. Got lots of brain things. So we can get into all this stuff and stuff that I did not even bring. So I am perfectly happy to see where the evening takes us and get us started fashionably late. Yes, Paul Disney. Always fashion. Always the science fashion. Okay, for those of you who are just joining in, who are not subscribed and do not tune in here on a weekly basis, we broadcast live. Normally it's myself, co-hosts Blair and Justin. Justin got a job recently, so he is not with us on our Wednesday evening sojourns, but Blair is usually here. She's out this week with a sick puppy. So send your love to Blair if you know Blair. We're hoping that her dog Sadie does okay. Send that love. And additionally, hope that science works in the land of the dogs. Wednesdays, 8 p.m. Pacific time is when we broadcast live on YouTube, Facebook, and Twitch. The podcast is found on podcast directories all over the place. Wherever you like to find podcasts, look for This Week in Science. You can find us by looking for This Week in Science on social media networks. Look for TWIS Science, TWISCIENCE. And our website is twis.org if you're interested in show notes and other fun tidbits related to the show. That's right. We have stories galore. And now, hit those like buttons, share. Let's get in the algorithms. Let's make sure science is not kept in a glass tower. Let's talk about it and make it something we're all excited about right here and now. You ready for the first story? Okay. My first story this evening, as I mentioned in the listing of stories, Epstein-Barr virus has been identified, and in previous years was identified as a potential viral cause or a trigger for autoimmune diseases like multiple sclerosis or lupus. And it's been this kind of loosey-goosey connection for a while. It's more and more evidence is being amassed. And so the hypotheses are getting tighter. And this week, a study is out that is, I think, very exciting. Researchers published in Science Translational Medicine, their study looking at how Epstein-Barr virus may be that actual cause or a driver of autoimmune disease. And in this case, they tie it to systemic lupus erythematosus. So in their study, they did use machine learning to do some of their analyses. But what they were really interested in is how Epstein-Barr virus infection actually impacts your immune cells. And what they determined is that by using a new strategy to identify these, what we'll call EBV, Epstein-Barr virus infected cells, they used RNA sequencing to be able to look at the immune cells, B cells, T cells, et cetera. And using RNA sequencing, we're able to see that infected B cells, and B cells are type immune cells of the body that are like the first kind of layer of in the first layers of defense. They are memory cells. And so you can have B cells that then talk to helper T cells, and then helper T cells go on to program and make more B cells so that your body's ready the next time it needs to fight. Anyway, when Epstein-Barr virus infects B cells, these immune memory cells, it makes the B cells basically do an antigen presentation on the outside of themselves. And so the helper T cells go, oh, what's the antigen that I am being presented with? And the helper T cells go and like grab onto that antigen that the B cells going, look, I have an antigen. This is a thing we should be fighting. Well, it turns out that antigen is from the nucleus of the B cell. So it is an antigen that is part of your immune cells. So the Epstein-Barr virus gets into the immune cells, and basically when it does its integration into the DNA and gets itself reproduced using the cellular machinery to make more EBV particles, in the process, it somehow, with RNA splicing, gets the B cells to put themselves on the outside and not the viral antigen. So then the T cells are like, ooh, look at this bad thing, this bad boy. We got to get memory cells and fight it. And so basically it starts a self-perpetuating cascade of the immune system highlighting your body, your own cells within the immune system as targets for attack. And so this is I think understanding this process is one of the, I think, very important aspects of being able to treat autoimmune diseases as we move forward. Because if we understand how Epstein-Barr virus is, it's probably one of many different causes of these diseases, but at the same time, if we can figure out how to stop it, if we can create a vaccine for it, if we can create a treatment that can just get in there and short circuit this particular immune system signaling cascade, then it can be, the potential is really great. And it also potentially this whole thing explains like why interleukins are so effective in their use for treating multiple sclerosis or lupus and other autoimmune diseases. Anyway, this is pretty big news. Understanding how this all works, it's essential to helping us live longer, healthier lives. And for those of us with family members and potential genetic predispositions to some of these autoimmune disorders, it's kind of, it's you start to breathe a little sigh of relief that there might be more treatments on the way. Maybe even cures. We'll see. Epstein-Barr virus is, and no, it's not the Epstein-Barr papers, the virus papers. No, Congress has nothing to do with these, Kevin Reardon. This has nothing to do with the newly sworn in congressperson in the Epstein papers that, we'll see if we actually ever see anything. This is actual science. Actual science. Oh my gosh. So, yeah, Epstein-Barr virus. You might know it as more commonly as it's a virus that causes mononucleosis, right? It's a virus, the kissing disease. Epstein-Barr virus is not something that most people are really affected by very strongly when they're infected by it. So Epstein-Barr virus, you can be infected by it and then just be like, oh, I felt bad for a little while and never even know it. But for those people who have genetic predispositions, or maybe there is also some tie-in with current levels of stress in the body or environmental conditions, or, whatever is going on, it has this potential to impact, to cause lifelong diseases that I think, interestingly, come into play later in life when there are hormonal changes, immune system changes, and additionally that tend to kind of ebb and flow, so that you can have a period of high activity of the autoimmune disease and then it goes into a remission again. And I think all of these aspects of these diseases, which has been so confusing for so many years, we'll see. I don't know. Kevin, I am not against like the political humor. Not against it. No. I mean, science is political these days. I mean, I've said it for years, but now there's no getting around it. So, yeah, well, if just please do not invite certain individuals to the party. Okay. Yes. Anyway, if any of you have questions about the Epstein-Barr virus or what's going on, there's a great article in The Guardian this week about this. There's a link to this paper in Science Translational Medicine, and I'll be sharing those in the show notes. Or you can ask me questions. I don't care. Well, I do care. I think it'd be great. Ask questions. I don't know if I can answer them. I'll do what I can. Additionally, some really good news. I love good health news. There's a huge clinical trial for a new drug produced by Novartis, and it's a combination drug that is, it's known as KLU-156, and it combines a drug called ganaplacide, which is, it's a new compound that was found by Novartis. They developed it. They screened over 2 million compounds for anti-malarial activity in the lab. They found this one, and they mixed it with an older anti-malarial called lumefantrine. And so the combination is KLU-156, and this combination drug cocktail It was studied in a clinical trial across Africa, 1,600 malaria patients across 12 African countries. They were treated for 3 days with either the KLU 156 or another drug that is called Coartem, and that is a more standard artemisinin. I can never say the drug. Artemisinin based combination therapy, and that is like the standard drugs that are used these days to treat malaria infection. And so artemisinin, or one of its derivative drugs, is they work really well. However, the problem is that Plasmodium falciparum, the malaria organism, this parasite, it likes to adapt and mutate and come up with new ways of outsmarting all of these drugs we like to come up with. It's kind of like the antibacterial issue that we've got. There's constantly a predator-prey fight going on. You come up with one therapy, it works for a while, and then the new adaptations are discovered by these biological organisms that allow them to continue to succeed, whether or not our treatments are used, in spite of our treatments, for sure. Moving on now with this massive clinical trial, the good news is that of the individuals that were given this drug, the 1,600 malaria patients, 99.2 of those who were treated were cured. And this is compared with 96.7% of the others, of the patients who received the artemisinin combo, the Coartem. And so this is hugely successful. 99.2 are being cured. That is absolutely imperative to be able to reach those kinds of numbers, because if you can't reach significant treatment levels, you're just going to continue to lead to potentially more adaptation by the organism to be able to get around it, because there is the exposure within individuals who go on to survive or don't, and the malaria can be spread. And so you end up with the spread of adaptations that are able to work around the treatments. One issue, though, even though this was incredibly successful, is that it made a lot of people vomit. So people weren't happy. They weren't doing well. They were kind of sick. And so a lot of people actually stopped taking it. Early in the trial, they would stop taking it and didn't make it through the 3 days of the treatment. So that actually was an issue. But they think, the researchers say, that this bitter tasting powder, they realized it needs to be given with a liquid, probably a sweet liquid or something. And they think that they can manage it better moving forward. And they did say that throughout the trial, they did seem to find ways to get people to not vomit as much, which I think is really good, because if you want people to take a medication, maybe it shouldn't make them vomit. I don't know. I think that might help a little bit. But malaria is spreading around the world. It is one of the, if not the top parasite killer, the killer of humans on the planet. It is around the world, global. It's global. It's spreading around the world as climate change continues to spread. And so we really need to make sure that we've got treatment options. So there is a vaccine that works kind of. It's not super, super effective, but it has been helpful with kids. And now with a treatment that could potentially be used in tandem with the older treatments, hopefully, if these treatments are used sparingly and with strategy, maybe we'll end up having them last longer so that we can actually find another treatment before they lose their ability to help us. But yet they're saying, Novartis is saying that they're hoping to get this into the hands of countries and hopefully patients within a year and a half. which is very exciting because hundreds of thousands of people are diagnosed with malaria or annually. Not just hundreds of thousands, excuse me. In Africa, the continent of Africa alone, 246 million cases. That's just diagnoses. 569,000 deaths. So anything we could do, save lives. I think it'd be pretty good. Let's keep saving lives, everybody. Yeah, and climate change. I mean, we're looking at the spread of these invasive infectious diseases as invasive organisms find new ecosystems and niches, right? So mosquitoes are moving, all sorts of insects, biting flies. Ooh, yay. we have ticks that carry Lyme disease that are moving into new ranges across the country. The funguses. Oh, the funguses. What fun we're going to have. And if you haven't read The Wanderers by Chuck, and I'm just blanking on his name, Chuck Wendig. If you haven't read The Wanderers by Chuck Wendig yet, that's a few years old, but I have to say that one's a good one. It takes a twist that was not expected, but then it's fun. I think it goes some neat directions. It's kind of like, I think it's kind of up there with, in terms of length, but in terms of the world building and the whole imagination of everything, I think The Wanderers is right up there with Stephen King's Stand. I think just this epic tale of people fighting against all odds, a hero's journey. Oh, hey, who knew? That's humanity. Let's go. Let's do it. I'm not whining yet, am I? I don't want to be whining at you all. I hope I'm giving you good news. Yeah, Paul Disney. Turns out people don't like to throw up. Who knew? Who knew? Oh, in other like really fun videos that are available out in the world, YouTube, if you are not a Zyla Foxlin fan, I just watched her video of flying a jet fighter for the first time, and it is the cutest, most awesome video. And yeah, she fully owns up to having motion sickness and vomiting. But I have to say she's inspirational. She's an amazing, it's like people go influencer. She's an engineer. She's a builder. She's a creator. She's a pilot. She's doing amazing stuff. And if you haven't checked out her work, I highly recommend what she's doing. She's really great. What else is out here in the world? Okay, I mentioned talking about invasive species, climate change, people moving all over the world. Not people, insects, but lots of animals, trees, everything's moving. And and this doesn't have anything to do with that at all, except this story in the Proceedings of National Academy of Sciences. The brief report is called Recent Immigration Raids Increased Student Absences. The researchers evaluated the claim of the connection between ICE raids and student truancy. It evaluates the claim. It used a daily time series of school absences spanning the current and prior 2 school years from 5 school districts serving communities subject to recent and unexpected raids in California's Central Valley. So this is very location specific. We're looking, in one highly impacted area, not across the entire United States. But they do find, and it says in their abstract, the results indicate that recent raids coincided with a 22% increase in daily student absences. with particularly large increases among the youngest students. So there is a massive impact. And if this can be related to other school districts, other parts of the country, nationwide, what we're looking at are impacts that are going to be negatively affecting the educational achievement of a young generation of people in America, people who are probably kids who are probably citizens, right? Born here, many others. Yes, we could say that ICE is having a chilling effect on education, but bump, which is really unfortunate. And you'd hope that there would be certain places in our communities that would be off limits, the places that are important for the functioning of our society, schools, hospitals, social halls, churches, community centers, grocery stores. I don't know. I feel like there is an impact. We're feeling the impact across the many layers of our society, but to know that the data is really supporting dimensions of the impacts on different dimensions of child development. So it's not just education, right? So if you have the youngest students not making it in, those students aren't learning necessarily their letters, their numbers, how to count, how to read. They're not learning the basic skills that are going to be the foundations for their ultimate educational achievement. So it's just the whole context and the situation is, it's really disturbing, and there's short-sightedness all around here. Anyway, the paper goes on to say the increased absenteeism it may also presage enrollment losses that add to the fiscal challenges faced by school districts. And this is after the pandemic and the pandemic flight from public schools. That also might reduce school resources in states like California, where state aid is based in part on average daily attendance. And the substantive implications of these increased absences are also likely to extend well beyond educational settings. The absences can be understood as a leading indicator of broad and developmentally harmful stress these raids create for students and their families. We don't know if the effects, if what effects are beyond absenteeism are occurring, and whether it's beyond California's Central Valley. That's an open question, but there is a need for continued attention to studying these immigration-related policy choices and promoting awareness of evidence on their implications. So we need to know a bit more of this. Yeah, I mean, it's a serious one. Not as much fun as supernovas, but I thought it was something that everyone should know about because education is essential. It is I use the word foundation a lot, but education is one of the foundational elements of our democracy, right? If people are educated, if they know how to read, if they know how to communicate, if they can critically assess information that is given to them, that is in their environment, that they come across, they will be able to behave critically as members of our social structure. Our social fabric relies on education. And so this is something very, very essential to what we're all dealing with and moving forward. Let's talk about supernovas. You want to do that, maybe? Yeah. Let's see. Oh, going back to Zyla Foxlin, also, like I said, amazing. Kevin Reardon is saying that Zyla got legislation passed to allow pilots to be allowed to have divorce counseling without losing their pilot license, which is fantastic. And that's something that I didn't realize years back. And it wasn't even that many years ago. It was just a few. I was traveling on an Alaska Airlines flight, and it was shortly after a pilot had had a mental breakdown, had taken psychedelics, was not flying the plane, mind you, but had taken these psychedelics, and it's basically self-medicating. And apparently, this is not something I knew, pilots cannot go to therapy. There's no counseling. There is no antidepressant medication that you can be on. There is nothing. It's basically as soon as you have mental challenges, that's going to impact your ability to fly, your ability to maintain your pilot's license. And so I think that kind of a mark, divorce counseling. It's not necessarily, I am having a mental breakdown, I'm having a mental emergency. It's trying it, but it's still counseling and could go on your medical record, which could be negative, right? So thanks, Zyla. That's cool. That's great. These are the things that we need. Make it easier for the people who take care of us and get us from one place to another, 30,000 feet up. Make it easier for them to be healthier, right? Why not? We should be, mental health is important. And just because you're having a crisis doesn't mean you're always going to be having a crisis. We got to do it. Yeah, anyway. Did anyone see the aurora last night? I right now my blinds are closed so that I can not have echoey reflections from my windows down here in my studio. So I don't know how the aurora is doing tonight. It's also kind of fall, so how many of you have cloud cover? But last night was apparently massive for aurora watching. People were able to see the aurora way down in like Southern California. I don't know if I can remember a time when that happened in my life, which is wild. Anyway. Yeah, going back to the pilot issue, yes, Paul, the intent is to select for only pilots who don't have problems, except it just makes pilots with problems hide them. Right. And maybe you don't have problems to begin with, but sometimes life happens, right? And you need help there. Yeah, New York City, you saw Dusk 108 is saying auroras in New York City. There's clouds up in the Seattle area. Kevin Reardon, there's clouds and rain where you are. Where else? I don't know. I'd like to go outside and see the aurora borealis, but it's not going to happen for me right now because I'm here with you. So I have pretty lights behind me. So I can imagine, I can pretend that I've got the aurora going on. Anyway, not talking about the aurora right now, although it is very exciting. But I was talking about the aurora. But next story, let's talk about supernova, supernovae, the plural. Two stories this week that I think are pretty interesting. The Swift Observatory, or the telescope, and the European Southern Observatory's very large, no, not the Swift. It's just fast observations. They moved really fast. I know that there's like a Swift observatory also, and so I think I got confused there for a second. The European Southern Observatory's very large telescope, the VLT, was focused onto a supernova, SN2024 GGI, that was detected the 10th of April, 2024. A researcher from Beijing, China, was landing in San Francisco, found out there had been a supernova, and sent a proposal to the European Southern Observatory to get approved. They got the VLT telescope in Chile to focus on the supernova just 24 hours later. So within, 26 hours later. So basically a day after the supernova had taken place. And if we can imagine, the size of a body that would go type 1a supernova and the amount of time that energy and the light is in the galaxy, the universe, and spreading outward from that point of initiation or ignition, 26 hours later is still so fast to be making an observation when we're thinking about something like this. And so this is the quickest turnaround on getting really high resolution measurements, let's see if I can say this word over again. They're using a technique with the VLT called spectropolarimetry. This is not just looking at the spectra of the stars. It's looking at the polarization of the light spectra arriving from that point source. And so the interesting, fun thing about this is okay, we have all sorts of hypotheses about a supernova happens. And if it's happening in this way, the shape is going to be like boom in whatever way. And so there are ideas that the shape looks like a football, that the shape looks like a frisbee, that the shape, that if different forces are predominant, that it would look a particular way and result in, and I'm just talking specifically about the type 1a supernova right now, which we use as a standard candle, which basically we're like, hey, if a star is a certain size, it's going to result in a certain luminosity, a certain amount of light coming out. But that luminosity, we see it as a point source. So like a spotlight or a flashlight turning on boop, and shining in your eyes, right? That point source coming from so far away, how could we ever figure out what it looks like, right? And so the spectropolarimetry takes into account from that point source little tiny deviations within it, as in the point source there are lots of photons, right, that are coming and being spun at just slightly different trajectories, velocities, and spins. So the polarity or the direction of spin of the light. And so it's not the so Kevin Reardon, it's not the polarization of a type 1a supernova that's different. It is the use of understanding the idea of polarity in light, that they are able to look at these little fine details within that point source that suddenly from the explosion, boom, light emitted, and that floodlight, that spotlight from so far away in our universe, we see it, right? That's what we see. It's like a bright star. Ooh, look at that. But there are little tiny deviations within the beam of light that has reached us, right? So the light we see has some variations within that area of space that is covered in our CCD detector or our radio array. And the polarity of that light and slight time differences were able to be used to be able to give an idea of what the supernova looks like. And so I'm going to show a picture to people who are watching online, and this is an artist's impression. This is not what it actually looks like, because I first looked at this and I went, what? How do we know that? And I have to say it's beautiful, but it gives the impression of like a big movie explosion, big bulbous releases of gas that are streaming out in directions from the top and the bottom. You would think, it's space, so what is top and what is bottom, right? But the data that the researchers got related to this supernova suggest that it breaks in vertical direction. So the two poles, basically, if you were to think of the North and South Pole magnetically in a star or a planet, that from those poles that there is the release of dust and gas and pressure from the supernova in those vertical directions away from the center. And then after, there is a flattening of the whole disk, or into a disk of dust and gas, as that supernova starts to impact dust and gas and particles that are around the star where the explosion took place. The supernova. Right. Patrick Pecoraro is asking, shouldn't it be spherically uniform? And that is one of the hypotheses that has been floated related to supernovas and how they look and why they look that way. And so it's not a cross section, but the idea is, we're getting only light that has been directed to us from, our perspective from this point in space. And it doesn't burst in a disk. It bursts more in a like busting apart from the top and the bottom, popping apart. And then the disk is not the burst. It's more the shock wave of the supernova impacting stuff around it. Anyway, in the graphic, whatever you call it, the description of the image that is here of this artist's impression, it's an artist's impression showing a star-going supernova about 22 million light years away. Supernova SN2024 GGI exploded in the galaxy NGC 3621. Using the ESO's VLT, astronomers managed to capture the very early stage of the supernova when the blast was breaking through the star's surface. Observing the breakout so early on revealed its true shape. The supernova broke out in an olive-like form. And this marks the first ever observation of the shape of a supernova explosion at this very early stage. And so, to your point, Patrick, why an olive? Why? I mean, why not a sphere? Why wouldn't it just be even all the way around? So what are the forces? Does it have to do with rotational inertia, right? That there's the stars orbiting, and maybe because of rotational and gravitational, rotational forces and gravitational effects, that maybe it's the spin that leads to this olive shape as opposed to spherical, right? Maybe it's not olive. Maybe we should call it an oblate spheroid, right? What about that, right? Is it a collimata, right, Paul? I don't know. Anyway, this is an interesting. It's one observation this soon after a supernova. So this is really the first and only of the data set at this early period of time. What the researchers say, though, is not that this is what they all look like. What it does suggest, however, is that there are some aspects based on their data collection. There are some aspects of certain hypotheses about supernova explosions that we can now count out, that we can get rid of some hypotheses of how the explosions occur, and that there may be some of the hypotheses that still need to be tested. We need observations for. We need to actually see more to be able to see what is really going on. But isn't it exciting? Yeah. Oh, hi, Trenton. Good to see you. Hi, Robbie. Who else is here? What else we've got? We've got Kevin and Paul, and yeah, no olive oil. Nope. And so, Paul, I think that is like your question here, I think is great. How much of the shaping is from magnetic fields? And that's another one that is definitely a possibility that I hadn't listed yet. But I think I'm not the supernova astrophysicist, so I don't know all of these details. But what I find very interesting about this is that the results have allowed them to, get rid of some hypotheses, but it's a pretty cool thing. There's let's see, we've got a video. Do you want to watch a video of the true shape of a supernova? Let's watch this. Go, a massive star went supernova. And just a day after the explosion was detected, astronomers observed its true shape for the first time. A star more massive than 8 suns. That's pretty big. Dies dramatically. Powerful explosion that completely disrupts the star's base. The very early breakout phase of the blast was only visible for a limited time. As the explosion expands, interacts with the material around the star, and its true shape gets distorted. And so that's where we usually see it, is after the fact, after the initial shape has occurred. So this short-lived early stage is what they were able to detect. And it's not a ball, it's an olive. Da, da, da. I hope they don't. How many supernova models? I am reading you, for those who are listening to this later, I'm reading the text of the transcript from the video that was in here. Anyway, I think that's an interesting one. How long till we know what was left over? Quite a while. Because, actually, I don't know. After a year, how much is left after a year? I honestly don't know. And I think that's a really interesting question. That's a great question. When do we get the follow-up? One of the great things about this particular supernova, however, is that it wasn't just visualized by the BLT. It was also visualized by amateur astronomers around the world. There were lots of other astronomers at institutions who observed it as well. So there's a lot of data related to this particular supernova. And so they will probably learn more and be reporting more as we move forward. So we'll see. That was pretty cool. There's another interesting supernova story this week. Apparently, there was a report in the monthly notices of the Royal Astronomical Society that it's raised a little kerfuffle in the astronomy supernova world, especially with respect to the question of how we can use supernova, type 1a supernovas as standard candles to measure expansion of our universe, as to whether or not it's accelerating or decelerating or just being boring and not doing anything at all. And as you know, if you've listened to or watched this show for a very long time, we've talked about the various measurements that people are making and the ways that they're trying to determine what's going on with dark energy and the expansion of our universe, and is our universe gonna end in the big crunch or the big bounce, or what's gonna happen? So this study in the Royal Astronomical Society monthly notices, its abstract basically says, we looked at a bunch of type 1a supernova remnants there, and the light that is coming from them or was coming from them, and we corrected for age bias. Basically, what they're saying is that as progenitor stars age and host galaxy mass evolves, and this also how that changes with redshift, that they say that there's a serious systematic bias with redshift in the supernova cosmology and how it's used. Basically saying that, well, we think there's a whole bunch of these supernovas that you're using as standard candles, and they're not really standard candles because you're not looking at the redshift right. So if you think about what they're saying and you don't ask all the questions, you go, yeah, they looked at the supernovas and they're saying this. So their new calculation that our universe is not accelerating. Wow, how interesting. It might be expanding, but not accelerating. And oh, that's great. Okay. However, there's a lot of supernova experts who are also coming out and saying, no. Oh, no, they didn't look at the right data set of supernovas. And in fact, they used an old, outdated supernova data set. And so what they have calculated isn't applicable to our understanding of the expansion of the universe. So there is a kerfuffle at the moment. And it's not really that big a kerfuffle, but it's interesting when you see, and now it's Blue Sky. It's not Twitter/X anymore at all. Blue Sky is where you go to see what the scientists have to say about each other's studies. But an astrophysicist responding to Phil Plait, the bad astronomer on Blue Sky, basically was like, yeah, no, none of us who really do this are taking this study seriously at all. But again, that's social media, and people say all sorts of things on social media. So I can't wait to see how this study actually ties in with the larger literature, larger body of literature, and what we end up seeing, because we really are still trying to differentiate the cosmic microwave background radiation measurements and trying to look at different measures to be able to understand, okay, here we are in space-time, what's happening around us. And we're at a disadvantage right now, being as we're just basically here. We have to go other places and set up other observatories and other arrays. And that's where we have to go. Kevin Reardon, good gracious, don't you go there. Oh, I like that. Paul Disney, yes. Cosmic yoga, the universe will end in the big downward dog. I think, what about child's pose? I mean, you do downward dog, and that's still like a strength and support kind of, that's something you're stretching, whatever. I mean, or cat cow. Ooh. I don't know. Yay. Oh, you're welcome, Pepsi Cola. I'm glad you're enjoying the space and physics stories. I do try. I really do. What I need to do is get, it was so great having Daniel Whiteson on the show a few weeks ago, and he had some wonderful things to say. We do need to get someone on who can really talk with us about the cosmic microwave background radiation, on the measurements, on what's happening with our supernova database, how we are looking at it. Because I seem to remember that we've had other studies looking at redshift. This is not the first time that anybody's done this. But anyway, we'll see. We'll see what happens from here. Anyway, they say, what do they say? They, in this particular study, using only supernova from young co-eval host galaxies across the full redshift range, they find a significantly stronger, greater than 9 sigma tension with the delta CDM model than that reported in the DESI papers, suggesting a time-varying dark energy equation of state in a currently non-accelerating universe. I find it fascinating. We'll see what happens there. I don't know where that's going to go. It could be the pose. The shrub pose, Brian. I love it. Cat cow can fix scoliosis and back pain, but can it fix the cosmic microwave background radiation discrepancy? I don't know. Yeah, Trenton, good to ask. How is everybody doing? I hope you're doing all well. I really do. This is This Week in Science. Thank you so much for joining me for another episode of This Week in Science. I'm Dr. Kiki Sanford, and this week it's just me and you, and I'm really enjoying talking with you about the stories that I wanted to bring to talk about. I do enjoy talking to my co-hosts a lot, but, it seems like I can fill the time, huh? If you are enjoying the show, please head over to TWIS.org. Click on that Patreon link, because Patreon is really how we are supported. We're listener supported. If you listen to us on some outlets, you might see some advertising, but that's through particular podcast directories, but not what we do specifically. So our podcast generally is just listener supported. That is my goal. My goal is to try to work with you and not be beholden to advertisers or particular interests for the news and the stories that we want to talk about. I want to talk about all of the science, and I hope that you want to talk about it too, want to learn about it and enjoy being curious. And if you do, help us keep the show going. Patreon is where you can support us directly, $10 and more a month. We'll thank you by name. I will try to thank you by name at the end of the show. I will do my best not to mispronounce your name. And additionally, we also have our Zazzle store. So as the holidays come up, merchandise might be great, right? If you want a pillow or a bath towel, a beach towel, all sorts of things, hats, mugs, great things. They make great gifts. There are things that are not just TWIS logos. There's also the pretty animal art if you have an art lover in the house. Wait, there's one. Blair has created amazing art through the years, and a lot of the products in our Zazzle store reflect that. And so I hope that you will take a look at our Zazzle store. Click that link that is at TWIS.org. That's where you find all that stuff, show notes, and more. Can't do this show without you. Thank you so much for your support. Oh, Trenton, thank you for being here. Yes. Thank you. I hope you all want to continue to be lifelong learners. It's so important. I mean, seriously, we keep hearing about these individuals who are living incredibly long lives, the super agers, right? And part of it, I think, is it's social connection, which we're doing right here too, because we're making we're having good social connectivity every single week, and intellectual stimulation. This is the kind of stuff that's going to keep you going. This is our engine for a long life, a long, healthy life. I'm not going to talk about the wine that I'm drinking, though. Robbie, thanks for being here. You're welcome. I'm glad that you're here joining us. Yes, I know. I miss my co-hosts right now, and I'm so glad that you are all here. Okay. I got more stories. Do you want to hear about a bee with horns? I think that's the next one up on my list. It's a cute little bee, actually. It's adorable. Published in the Journal of Hymenopteran Research, which I love that there's a whole journal of Hymenopteran research. Researchers have identified and reported their discovery of a new species of bee. They found it in Western Australia's goldfields when they were studying a critically endangered wildflower, Merianthus aquilonarius. This particular flower only grows in one particular area in Australia, which I think is so amazing because basically here, I have to, how can I get this in? There we go. I'm putting my pictures different places so we can see all these things. But this host plant species is critically endangered. It only grows between two towns in a region of Australia known as the Bremer region. And this Bremer Range region has these towns, Norseman and Hayden. And this wildflower only grows there in the whole world. This is the only place that it grows. And so they're there looking at the flowers, right? They're there going like, hey, what's up, flowers? Here, let me show these pretty little flowers. They're pretty little flowers. These plants, they're very delicate little flowers on a larger shrubby kind of bush. So this plant, it's beautiful. Here's a map also of Australia. So you can see this little area just east of Perth, Western Australia. It's kind of dryish, but it's in the southwestern quadrant of Australia. Never eat shredded wheat. Yep. And it is that's right, Trenton. It's an incredibly rare flower. Here. Oh, I didn't put this up here for you. Here we go. And I need to get that. Look at me. I can make all of the things go like I usually do. So this map of Australia is showing where this flower lives. Shows it's in this little teeny tiny region. This flower is this cute little flower. It's an adorable little flower that lives there. And this is the only area in the entire world where it grows. So they're there looking at the flower. And while they were there looking at the flower, they discovered this species of bees. And they discovered the species of bees during the mass flowering of another flower, a eucalyptus plant flower. So the Eucalyptus lividea, it has also a limited distribution and a short activity for flowering and its season. And it's also associated in this critically endangered region where this Merianthus flower is growing. So we've got two Merianthus critically endangered. We've got this other eucalyptus species that is mass flowering during a short flowering event once a year. What? This is crazy. The possibility of like identifying this insect is probably so ridiculously small, but because they were there doing the work at the time, here they go. They found this pollinator, right? And so lots of places are undergoing pollinator crises at the moment because of climate change, because of insecticides, pesticides. We've got issues where you don't have the pollinators showing up where they need to be anymore. And so in this particular situation, what they have done is they found a new species of megachile, Hacariapsis. So they have males and females, and the females have horns, which is really interesting, right? So these females, they're a little horny. And yeah, I did go there. I really did. So the females, they're cute. They're decorated. They've got horns, but why do they have horns? We don't necessarily know yet. But they have been able to show that the females have horns, the males don't. The prominent horns on the clypeus, it's a deeply depressed with a medial ridge. And this species visited the Merianthus and also the other species, the Eucalyptus. And so this is occurring only in this little tiny range of Western Australia. And so they're hoping to promote more research into the horny bees, because that you got to tell people about things that you find so that people can study them. Study the horny bees. The males are cute. They're gray bees. They're not the brightly colored bees that we think of when you think of the yellow and black stripes. These are more of a gray or a white and gray coloring, maybe a little brown on the rump of the thorax. Trenton is asking, what is an endangered region? And does that mean there are lots of endangered plants? And yes. So in that area, probably a result of climate change and changing whether or not there's fire or human encroachment or what's going on, that there are lots of plants and animals that are endangered. Specifically, however, these flowers are incredibly, they're cute little flowers. So cute. And they are visited by these sweet little bees who have never been described before. And the ladies have horns on their head. And we don't know why. They are really cute little horns. I really want to know what they're for. Why? Why do you have horns on your head, little lady, little bee? And of course, as we think about males and females, these are not the queens. So when we're thinking about male and female in terms of Hymenoptera, of bees, that the genetics are, it's a little bit different because you've got the queens, then you've got females, you've got male worker drones. There are, there's a lot more going on in the genetics of sexual differentiation than would, than our limited descriptions that people argue about because they have political reasons. Anyway, yeah, they're so cute. I want to hang out with these bees. They're adorable. The females are much darker in color than the males, actually, and I would love to know more about them. I think it's very fun. Anyway, okay, we'll move on from bees. This is the only animal story I had this week. Did anybody here have any good animal stories that they want to share this week? Let us all know, because Blair's not here, so that was my animal corner. That's what I'm giving you. Oh, Trenton Moody is saying anyone who likes botany should check out Crimes Pays, but Botany doesn't. Crime pays, but botany doesn't. It's true. I have not heard that, so thank you for that. I will definitely check it out. Oh, is Snake here? Hello, Snake. I hope you're watching the show. Welcome. And is friend Snake an actual snake or a person with a nickname Snake? I don't know. Because that makes me think of Snake Plissken. Right? Can we get a holler for Snake Plissken? Beware of snakes. Does it have to be snakes? It had to be snakes. Last week it was spiders. This week it is snakes. Let's see. Oh, okay. Here, this is another aspect of this bee story that's super cute. The article from Phys.org, Pensoft Publishers. They've said here in an interview from the researchers that the name was inspired by the Latin for light bringer, and that the name of this new bee, the horns and everything, inspired its name Lucifer. So this new Megachile Hecariapsis Lucifer is the whole name of this very sweet little bee, and the researcher says the female had these incredible little horns on her face. And when I was writing up the new species description, I was watching the Netflix show Lucifer at the time, and the name just fit perfectly. It's fantastic. This is the first new member of this bee group described in more than 20 years, which is super amazing. Okay, let's talk brain stuff. I have a few brain stories, and then I'm done for the night. I won't keep you here all night. Oh, yes. If you are a bird watcher, Patrick is talking about a bird snacking on a snake like it was a rope of licorice. There are many birds that you'll catch doing this. I love shrikes. You'll catch them eating snakes and lizards. Additionally, very often, if egrets or egrets are very often snake eaters, or there's lots of birds are generalists and will eat what is available at a particular time. So it's like, oh, a snake, and they will eat it. Yum, 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 yum. Exactly. All right, brain news. Sometimes I feel like I am a little, I'm not consistent from different times of the month. I'm not going to talk to my husband about this really right now. I mean, and you all might have your own observations from seeing me week in and week out on this show. But researchers have just published in Nature Neuroscience their work looking at the influence of estrogen on dopamine neurons. So how does estrogen actually impact dopaminergic release and the impacts of dopamine in learning? Because dopamine is very, very highly integrated into our seeking and learning behaviors. Because if you have a reward, dopamine's like, yeah, I'm gonna get it. Okay, got the reward. And then start over, look for something else. So dopamine is the thing that keeps us going toward a goal, toward a reward, toward something. And when you're learning something, it's like a surprise, or you are reading and training or studying. The dopamine is the thing that keeps you going. And it's the dopamine hits that keep pushing you to learn new things. And you want to keep that coming. But there have been questions related to the variability of estrogen release and other hormones in the body with respect to how they influence the neurons that release dopamine that are involved in our reward pathways. And so learning, cognition, and other behaviors. Over and over again through the years, there's lots of research that's like, yay, yes, memory up, memory down, or these behaviors, these moods, these things change based on changing estrogen and progesterone levels throughout a woman's cycle. So this study manipulated estrogen in rats, not in women. This is in rats. And they were able to show in these laboratory rats, which were trained to basically go through a puzzle to go get water. And the dopamine is released in the learning how to get through this challenge to get to their water reward. Okay, so if you have more estrogen, how do you do learning the task? Less estrogen, how do you do learning the task? Because the estrogen, the hypothesis was that it would either increase or decrease the amount of dopamine that's being released by the neurons in the brain. And that's exactly what they found, is that when estrogen was high, that led to higher levels of dopamine neuron activity. Estrogen boosted dopamine. And so dopamine had its impacts on the brain's reward center, making reward signals stronger. So learning and then memory, right? Your learning is stronger with higher estrogen. Estrogen lower led to less learning. And so potentially there are ways that we can understand this. So if you're taking hormone replacement therapy where you have a consistent amount of hormone at various times during the month, then that's going to maybe replace these pulses or changes that would impact your learning at various times. Alternatively, we can think that if you're not taking something external that's going to be influencing your hormones, right, that maybe you can work with the body's moods, right? The changes through the month, understand when estrogen's high, when it's low, and schedule around that. What are the best times to be studying? What are the best times to be resting, right? What are the cycles and how can you perform optimally? But this is really starting to get at, and there are more studies to come related to this, but get at the specific link between hormones and our behavioral neurons in the brain. How do hormones actually change cognition? What is the link? And that is something that the mechanisms are things that we are not entirely sure of. And so this is another piece in the puzzle. Kind of cool. Dun, dun, dun. I love studies like this. I think they're very interesting. For those of us who are getting older, we often worry about where our memory is going to go. What word was I going to say 2 minutes ago? I don't even know anymore. Don't know what I was going to talk about. There is a compound called nicotinamide adenine dinucleotide, the oxidized form, NAD+. NAD+ is a compound that has been shown to potentially have some real positive benefits for memory, for brain function, for how the body, for keeping the brain and your neurons healthy as you're aging. And I know that it's been an interesting kind of push-pull with the FDA and what's approved and what's not. And NAD+ and NAD have kind of gone into and out of favor as supplements. And I'm not, this isn't about supplements necessarily. What this study is indicating, however, is published in Science Advances. The researchers looked at how this metabolite, it's involved in energy metabolism, helps the neurons, usually declines with age, right? So this is also a period of time as you age, your mitochondrial function decreases, neuronal function is not at its top form as you age. So how can we potentially, or would it be worth it? Is it important to maintain NAD+? Can it help things? They looked at using NAD+ to augment. So in this particular study, I think they gave it to Alzheimer's patients, and they found that when NAD+ levels were increased, it corrected damage caused by the tau proteins. And so Alzheimer's cognitive deficits, the memory deficits that are prevalent within Alzheimer's patients, were reversed. And so this is a very exciting study in which this hasn't been done before, right? That they haven't shown NAD+ to have this particular pathway of action. And so this action is RNA splicing. It's regulated by a protein called EVA1C. And the EVA1C plays a role in the RNA splicing. And so when the splicing happens, then you can get multiple forms of a protein. And so you can get the good forms or the bad forms. And when you have more NAD+, EVA1C fixes its mistakes more often. So you don't have as many bad splices and bad isoforms of or dysregulated proteins with the RNA splicing that happens as a natural part of metabolism. And so this can improve the function of all sorts of genes all over the place. And what they discovered is that when they got rid of the EVA1C gene, they lost all of those benefits. So they found that with people with early onset Alzheimer's disease, that EVA1C levels are significantly reduced. So the idea is that could we supplement and make it a regular treatment to give people, find ways to either fix the EVA1C gene or to increase NAD plus within the body naturally? Can we keep it from declining in its prevalence within the body naturally? Because if it's there, then it continues to have all sorts of great effects like boosting EVA1C, which helps to stop cellular gene splice mistakes, which makes your proteins work better, which makes your memory work better, your cells work better. I bet it had, I got to get myself some. I don't know. I'm not into supplements, but this might be one. I'm not sure. Yeah. So NAD plus, this is one I'm keeping my eye on. I'm not one to jump into supplements. I think supplements for the large part are oversold and their benefits highly overstated. And so, the question also is when you have an extreme case like Alzheimer's disease, where you have massive damage to cells of the brain, is that the state where reinstating some activity of, say, EVA1C or NAD+, is that the place where the benefits are seen? If you are aging naturally, where just slow decline of the functionality of things, does it have the same benefit? And these are the questions. It's like the cost benefit analysis, right? Is what's the downside to having an increased NAD+ level? And if there is no downside, is there an upside? Like, does it actually do much good? Can you take it prophylactically to keep from ending up in a situation that's like dementia or Alzheimer's? And these are questions we do not have the answers for. Patrick Pecoraro is going to die during the heat death of the universe. I think that's a great plan. It sounds like you're sticking around for a while. I'm there with you. Let's do it. All right. This is my final story. Did I have two more? Okay, two more really quick. So one interesting one, this is a preprint. This actually has not come out published yet. Researchers have published this in the Sci archive. So this is psychology papers in the an archive for that. It's preprint. So they're getting peer review of a sort before they submit it for publication. The researchers, my area of study when I was in graduate school was the hippocampus. The hippocampus of the brain is it's important for memory, right? It's the first part of the brain that develops to allow communication with other areas of the brain for storage of memory, for connecting memories, for connecting all of the information that is essential for what we call episodic memory, the who, what, when, where aspects of memory. Because it's not like, oh, all of that memory is over here in this one little pixel in my brain, and that it's just a librarian goes and pulls it and pulls the memory down. No, it's like, it's a whole symphony of neural signals related to each other in time and space through perception, but coded into our neurons. And our hippocampus is the conductor. It's a librarian conductor. And so the interesting question they were asking is, as we develop And we know different stages of life where we're able to remember different complexities of memory. How does that also tie into brain development and that what the hippocampus is tying into more solidly to be able to tie different memories together? And so in this paper, they bring up this idea that there's a 3-stage framework for the development of temporal memory, resulting that from this kind of change in the ability of the hippocampus to talk to different parts of the brain. So it starts out with young kids not being able to remember, contextual stuff across time and space. It's very event specific, very local. The way they remember things is very different from how an adult remembers. And so that's really because the hippocampus is immature. Functionally, it is not able to connect with lots of other areas of the brain. The frontoparietal cortex is still developing. And so because all these areas of the brain are still working, it underlies this kind of immaturity and inability of young kids to substantially and consistently tie locale and events to time. So temporal memory is it's hippocampus only, really. And then as they get older, kids and adolescents, they form and reinstate global knowledge of temporal regularities. And this is a result of enhanced interactions between the hippocampus and the lateral frontoparietal cortex. So the hippocampus in the middle suddenly is talking to this frontoparietal cortex and able to start kind of going, okay, time, place, paying attention to more things, able to place things and tie one thing from maybe today together with something similar yesterday, right? Oh, this thing that we did, this is the same as that other thing. And so as adults, we have hierarchical knowledge, details, generalities, new environments, old environments. We tie things together in context. There's all sorts of things. And this is mature hippocampus, mature lateral frontoparietal cortex, and medial frontoparietal cortex. And so this is the whole brain, actually, like that's the attention part of the brain, the frontoparietal cortex, talking much more consistently with the hippocampus and those connections being stronger that allows memories to form in the way that they do. And the big question from all of this, now that I've gotten through all the sciencey science stuff of it, that they're asking is that when we are talking to kids in a legal perspective, asking kids to specifically identify time and place when something happened to them or that they saw or that they experienced, can we talk with them about time-based context if they don't perceive time or remember based on time in the same way that adults do? So the fact that episodic memory is basically an adult thing that allows us to segment experiences into discrete events and everything. This is critical, but we have a very, our hierarchical temporal knowledge is very specific in a way that is not possible for kids. And what they say in this paper is children often struggle with both recalling temporal details and forming knowledge of temporal regularities. But we often ask them to pull up memories in a way that adults would have to. And so is there a better way? Understanding that kids' brains develop differently. They experience and remember time differently than adults do. How can we work with them better? make experiences work. Good night, Kevin. Thanks for enjoying the show tonight. Thanks for joining. Loved all your comments. And then finally, last question from me to all of you is, what do you think about these brain organoids, right? As there's been a few opinion articles and different papers being written recently about these little clumps of brain cells grown in dishes that we are using to, grow brain tissue, astrocytes, neurons, blood cells, other supportive cells, like multicellular balls of cells that are tiny but three-dimensional structures and not flat plates of cells in dishes. These are three-dimensional networking balls of brain cells. Are we worried about the ethical considerations of potential sentience in dishes? Yep. I think they're great too. I mean, so far, I think that it's not an issue whatsoever. My question is like, at what point, like, do we stop? How big does a clump of brain cells have to be? And should it be connected to sensory organs or sensory cells? Because I can't help but think that sentience is an emergent property of not just the brain cells, but the body, the perception, the sensory aspects. You can't have one without the other. Yeah. Pepsi Cola. Yes. We're all hoping that our brains outdo the brain organoids. If you can beat a dish brain, you're doing great. 10 years to go. Let's see where it goes. Yeah, I guess what I'm questioning is how I'm reading these ethical quandaries out in the science media and what's being written, how much is being written about them, and how sensational they're making it. Is it just bringing up topics and it's fine, or are people really concerned about this? Like, really? Do we really need to be concerned about the sentience of dish brains? I'm going to leave you with that for the night. Sleep on it. Sleep on the dish brains. Tell me what you think. I want to know how you feel about the rights of little brain organoids. Do they deserve consideration? Mm-hmm. I don't know. Brian Burwell says maybe Petri dishes is where Boltzmann brains get their start. I mean, it's true. They got to start somewhere. Oh, no. Fada. I was beat out for a computer software engineer job by a brain organoid in a dish. I certainly hope not. We should not be at that point yet. Computer software engineer, if you're talking about a large language model, that's happening more and more often. It's really upsetting. Yeah, Pepsi Cola, you feel it's similar to AI, overly sensational. We do not understand our own consciousness and beginning of life. We're nowhere near intelligent enough to create a brain like ours, nor Terminator. Yeah, and that's where I'm as well. I think that it's good to bring issues up to the public, and I understand that you have to bring things up well in advance to think through possible outcomes, right? Possible paths, possible outcomes. It's a great practice, right? It's a good engineering practice as well. And philosophically, it's great to think things through. Go, where is this going to go, and where do we want it to go? Have we considered all the angles? And so, I think sometimes these articles that are just the philosophical discussion within science, whatever, they get, overstated and a little bit sensational when it gets into the public, because then it turns into our brains in dishes alive and sentient. Well, yeah, they're alive. They're cells. It's alive. Oh, they're not going to reproduce or anything, but it's a very interesting. It's interesting. It's that interface between science and society, right? And we should be having these discussions together. And it should be a situation where people are kept abreast of the philosophical questions, right? What are we okay with and what are we not okay with? I think it's essential that people are a part of the process. But at a certain point, if it's sensational, then people aren't really a part of the process. They're just being played with and manipulated. Oh, outrage, or ooh, wonder and astonishment. And yes, I do like to play the sensational fiddle every once in a while, but I always try to bring it back to reality when it comes down to it. What do I understand? What do we understand? What questions do we have? Because it can be fun. It's good. It's great to have fun, but we got to be serious every once in a while. I'm seeing Fada respond to okay, so the brain organoid in a dish had 2 PhDs and a half a dozen registered US patents. Well, I see where you're going, Fada. Blah ha 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 ha. Did he had 2 turntables and a microphone, maybe? Because it's where it's at. Have we done it? I've done it. I've gone through all my stories tonight, and I did 2 hours by myself with you. Thank you for joining me. I can't believe I just talked this much. I guess I needed it. I hope you did too. Thank you so much for joining me for the show tonight. I always enjoy chatting about science, and I hope that that some of this was curious for you and that you enjoyed it as well. I want to say thank you to those of you in the chat room who are here with me hanging out, because I couldn't have done this without you. I mean, really, it was us tonight. We're hanging out talking. It's just us. So thank you for being here. Twitch, YouTube, Facebook, all your comments, I really appreciated them. Fada, thank you so much for your work on social media and doing the show notes. It just is so helpful. Rachel, thank you for editing the show and getting it posted to our website. And Identity4, thank you for recording the show. And as always, thank you to the TWIS Patreon sponsors. Thank you to Aaron Anathema, Adam Mishkan, Alan Viola, Ali Coffin, Andrew Swanson, Artem, Arthur Kepler, Bob Calder, Bob Coles, Brendan Minish, Brian Carrington, Chris Rosniak, Christopher Dreyer, Christopher Rappin, Craig Landon, Craig Potts, Dana Lewis, Daryl Myshak, Dave Wilkinson, David E. Youngblood, Don Mundis, Donathan Styles, a.k.a. Don Stylo, E.O., Eden Mundell, Eric Knapp, Flying Out, Fredis 104, G. Burton Lattimore, George Corris, Greg Briggs, Jack, Jason Olds, John Atwood, John McKee, John Ratnaswamy, Johnny Gridley, Ken Hayes, Kent Northcote, Kevin Parachan, Kevin Reardon, Kurt Larson, Lon Makes, Lauren Gifford, Marjorie Gertz, Mark Hessenflow, Noodles, Patrick Pecoraro, Paul D. Disney, Paul Ronovich, Paul Philip Shane, Pierre Velazar, B.I.G., Richard Badge, Robert Norland, Robert Farley, Rodney Lewis, Rudy Garcia, Remy Day, Sean Clarence Lamb, Shubrew, Stephen Alberon, Steve Leisman, a.k.a. Zima, Sue Doster, Teresa Smith, Tony Steele, and Vegard Schefstad. I don't have anybody else to clap, but thank you all for your support on Patreon. We cannot do it without our Patreon sponsors. You are essential to what we do here at TWIS. And if you are interested in helping us, head over to twis.org, where you can find information about the show, where you can find show notes, And you can find the link to our Patreon and our Zazzle, where you can support the show all on your own. And if you do $10 a month or more, I will probably mispronounce your name at the end of the show. Come on, don't you want that? Be a part of that, of my tongue twister. I try. I do try. On next week's show, hopefully Blair will be back and we will be talking together about more Blair's Animal Corner stories. And I do hope that you will be here to join us next Wednesday at 8 p.m. Pacific time-ish, broadcasting live from our Twitch, YouTube, and Facebook channels. If you want to listen to us as a podcast, not live, you can search for This Week in Science wherever podcasts are found. And if you enjoyed the show, get your friends to subscribe too. For more information on anything you've heard here today, show notes and links to the stories are going to be available on our website, TWIS.org. You can also sign up for our newsletter. I swear it's like right there. It's going to happen. I'll get Blair to do it eventually. We love your feedback. And if there is a topic you want us to cover or address, a suggestion for an interview, please let us know on one of our social media accounts. They're out there. They really are. I try to check them every once in a while. Send us an email, though, if you really want to get in touch. Just don't put random things in the subject line. Make sure TWIS is in the subject line so that your email doesn't get spam filtered into an olive-shaped supernova that I didn't look at. Oh, I missed the olive pit. Oh, dear. We look forward to discussing science with you again next week. And if you've learned anything from the show, please remember, it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.